The second worksheet deals with promoters. So when you open this one, it explains what a promoter is. It is a region here before the gene, around your beginning part. Your transcription start site is in here. We have typically thought of them as TATA boxes in bacteria. There can be others, especially in eukaryotes. There can be many, many different promoters or promoter sequences. You need to read through this. Promoters are different from enhancers. Enhancers are much further away from your gene than the promoter. This kind of explains what a promoter is. It's a region where the RNA polymerase will bind. We discussed this previously in the semester. All right, CPG islands are often in this region. Your transcription start site will be in this region. All right, so we have two types of promoters here. There are sharp promoters. A sharp promoter is a type of promoter where there is just one transcription start site. There is just one place, one very specific place for that gene to start, and that's going to make this a sharp promoter. It's when you have one place for essentially the gene to start. Sometimes, however, you have something called broad promoters, where there are many transcription start sites. These are often found in regions with high GC. So if you have a lot of GC, you might end up with several possible places that this transcript could be initiated, the transcription start site. So RNA polymerase may bind at different places at different times, and given the GC content here, it might be different in different individuals, might be different even in the same individual but in cells due to something called methylation. We haven't talked about that, but the idea is that a gene doesn't have to start in one particular place. We used to think that they were all sharp. We now realize there are broad promoters. Metazoans are flies. All right, that's essentially what we're talking about here. And I've given you quite a bit of information about flies here. A core promoter for a fly is a little different, so read about these. You don't necessarily have to do them. What you're going to have to do is find the sequences that are associated with each of these. TATA, you understand. These are listed here. There are certain sequences that are related to this, and you're going to have to find these sequences in a larger DNA sequence and find out what the nature of a promoter is. All right, so here are the elements. For instance, an INR has a sequence of YYA NWYY between the 5' and the 3'. You're gonna learn about this in your sequence. We already learned about what Y and W are when you don't have to use your slashes here. The IUPAC names for those. That was in the previous video that I explained that. So go back to that and make sure you figure out what these sequences are. Now, a consensus sequence is a sequence where this was found to be a promoter in several organisms or in several different genes. It's a place where a protein will bind and usually do something. And it doesn't have to be one specific sequence. Often several different sequences will have the same function in a promoter region, and there is some variability, and basically this is telling you that in general the consensus is it's like this. It doesn't have to be exactly one way or the other. It could be a C or a T, for instance. That's kind of telling us here what to look for. And you don't look for just one sequence. You're going to have to look for a sequence that has some variability in it. For instance, the TATA box is simply not TATA. It's TATAWAAR. So you have to go find out what the W and the R are. On that previous video, I showed you where to find that. You go through and you will have to look for a TATA, but then you have to look for these variables in there as well. So just keep that in mind as you're doing this. So you'll search for these sequences in this paper. It's a little long, but if you'll read through it, it's actually shouldn't be a hard exercise. It's kind of like a crossword puzzle. you're going to be kind of looking for, or one of those search puzzles where you have words within a big list of I forget what those are called, searches, word searches within a matrix of letters, and you find words inside of it. Basically, that's what you're doing here. You're finding these like words within the longer sequence. So you're going to be doing what we call sequence analysis here. So you need to read through this, find out some of it. Some of this is more important. Here is your assignment down here. You're going to do Drosophila, so that's flies. You're not going to be searching for human. You'll be searching for through flies and find a gene called the heat shock protein 70, or HSP70. You should be able to just search for HSP70 in the Drosophila genome, and it should give you a gene, and you're going to find the region near that gene, near the beginning of that gene, that has these sequences in it. You're going to find out which sequences are there, because finding promoters helps you understand how this gene is regulated. All right. Notice that the graduate students will have to do this for not just HSP70, but for HSP26. And that should explain that worksheet.